But cancers need to do more than just proliferate to become a cancer. They need also to evade something called programmed cell death, which is something that occurs in our body every day, something like 20 to 60 billion times. And it's a process that our body uses to remove cells because they're damaged or they're dangerous, like a cancer cell. Now, this is a new way of thinking about cancer. And the discovery that was made was that a group of proteins called the BCL2 proteins were responsible for keeping the cell alive when it should die. And cancer cells stop dying by upregulating these proteins. Now, it became apparent at that stage that if you could make a drug that stopped these proteins from working, you'd have a drug that could reinitiate death in cancers. So drug discovery programs were initiated by a number of groups around the world, including us. And over 10 years ago, we started trying to find compounds that could stop these proteins from working. That started through medicinal chemistry colleagues doing high-throughput screening and finding starting points. But a key piece of information that you need to have to develop these drugs is understand the way the protein and the drug interact so that you can enhance that drug, you can make it a better drug. And that's where I come in. I'm a structural biologist, and we use a process called X-ray crystallography to understand how proteins look. And to do this, we need to use the synchrotron, which is, to us, an awesome piece of machinery. For those of you who don't know what the synchrotron is a facility out at Clayton, the size of a football field, and essentially what it does is it produces intense light across the whole spectrum. But we use it for using the high-intensity X-ray beam to do our experiments of crystallography. Essentially, we take our protein of interest, we grow a crystal out of it, and then we shoot it with the X-ray. And by doing that, we can see at the molecular level the atoms that make up the protein. And importantly, we can also see drugs that are bound to it. And once we can see that, we're able to take those drugs and improve them so that they become high-affinity inhibitors. We use the synchrotron to develop these compounds to a high-affinity inhibitor, and at that point, companies started getting interested in them, and particularly 2 companies in the US got interested, and we initiated a collaboration. That collaboration was based on the compounds that we had, the fact that we could see the way they bound to the protein, and the fact that we had regular access to the synchrotron so that we could keep improving them. That collaboration has resulted in a number of outputs. Over 50 publications have come from that collaboration, 10 patents protecting the work that's come out of it, and over $20 million of reinvestment into the research that's going on at the WEHI. There's also the potential of ongoing milestone payments and royalty payments if the compounds that we're developing get into the clinic. But more importantly and more excitingly, there's a particular compound that's come out of this collaboration called venetoclax, which is currently in phase 3 clinical trials here in Melbourne and elsewhere in the world. And the clinical trials that compound is being used for is for a disease called chronic lymphocytic leukemia. Unfortunately, 1,300 Australians get chronic lymphocytic leukemia, CLL, it's easier to say, CLL every year. And of those, 350 people succumb to that disease, unfortunately. So it's in need of new treatments. And this new treatment in these clinical trials are really quite promising. We're hoping that these clinical trials will be completed in the next 12 to 18 months, and then this compound will go into the clinic. So it's a nice story of starting from very basic research discoveries all the way through to a drug discovery process to a final outcome that is helping patients out there who need help. The synchrotron was really important in this development. In particular, our regular access to the synchrotron rapidly accelerated our ability to develop these compounds. Previously, we would go to international synchrotrons, and we might get there once every 6 months. Now we can get there every 3 to 4 weeks, which immensely increases our capacity to do this work.